Few will forget the pandemic and the loss and disruption it caused. But behind the scenes, scientists were doing serious research right here in Trinidad and Tobago. One of those scientists was Professor Christine Carrington, a virologist and UE lecturer who led a small team of researchers sequencing samples of the SARS-CoV-2 genome. Professor Carrington is the Anthony N. Sabga Awards Caribbean Excellence Laureate for Science and Technology. The COVID-19 pandemic started for virologist Christine Carrington, her husband Axel, and their 2 children, like it did for pretty much everyone else.
Initially, when I saw it, I remembered the original SARS coronavirus, which we managed to contain. You did have quite cases, and you did have deaths in Hong Kong, and you had deaths in Canada, but it wasn't a pandemic. And so when I saw it first, I thought, okay, they've seen it, they'll get it in hand. But after a while, it became clear that that was not what was happening, and it was alarming.
But as a researcher whose expertise is virus evolution, she quickly realized that she and her team should be answering important questions.
Understanding the evolution and the genetics of these viruses can give you information that is useful to public health. You can work out things like when a virus emerged and where it came from, you know, how fast the virus population is growing, things like that. If cases are linked or not, things like that you can work out. And so we felt it was important for us to sort of have a baseline of information about SARS-CoV-2 in the Caribbean. Hi, Nikita.
Hi.
These are the samples, the COVID-19 samples that we got from TPHL on Friday.
Yes, so I'm prepping those.
In April 2020, they came up with an idea to sequence the genomes of a few hundred COVID-19 samples taken from across the Caribbean.
We were only going to do like 800 samples. We applied for money from UE to be able to do that. Not a vast sum of money, it's 100,000 US. Which is not a lot for research. My postdoctoral researcher at the time, Nikita Sahadeo, she was the person tasked with, you know, trying to get this sequencing implemented and everything.
Christine and Nikita settled down to work, hoping for a relatively leisurely pace of research.
It started as a simple sequencing project to establish the capacity. You know, I had just graduated with my PhD. I was just very excited to be doing something new and trying something new and getting back into the lab and learning.
On December 5th, I think we did our first 5 samples. So that was the first time we had a whole genome of a virus had been sequenced in Trinidad and Tobago. We did 5 Trinidad and Tobago samples. And we were very happy, very pleased with ourselves, and we thought we'd settle down to, you know, relaxed pace of research, looking at these viruses, getting samples from CARFA. And then about 2 weeks after, the first variant of concern emerged in the UK. That was the Alpha variant. And at that point, the WHO and Pan American Health Organization recommended that governments do surveillance for these variants of concern and do this genomic sequencing.
Wuhan in China now on lockdown.
Tonight, coronavirus exploding coast to coast.
Desperate measures to try and contain the deadly coronavirus.
The Minister of Health confirmed today that Trinidad and Tobago has recorded its first case of the novel coronavirus.
Today in Trinidad and Tobago, we have a population that is now scared.
Variants of concern were popping up all over the world, and the chief medical officer called on Christine and her team to start sequencing genetic samples, not only from Trinidad, but from other Caribbean states as well. It meant a dramatic increase in the volume of work.
Suddenly, we were getting samples from everywhere. The chief medical officer contacted me and told me, you know, he'd like a weekly report on the sequencing that we're doing and things, and it all changed. it was one person. It was Nikita in the lab. I had teaching and admin and other things to do. I don't get in the lab very often, unfortunately.
I mean, the only thing she didn't do is come into the actual wet lab and do wet lab work because she couldn't. But I think if she could have, she would have. But she was pretty hands-on everywhere she could have been, which was great.
So Nikita is in the lab, setting this thing up, running these samples, doing every single step. And it's quite long, tedious, and then they trickled in first, and then it was constant. So
sometimes, yeah, from 10 a.m. to 11 p.m., I would just be in the lab.
I think one of the hardest parts was, there's a lab work, but then once you have this genetic data out, you have to understand what it's saying. And it's a lot of data, it's big data. And there's a lot of bioinformatics, computer work that goes in to taking this raw data and turning it into something that we understand.
It was different to any of the other kind of work we'd ever done before, actually having to be so careful and the reporting of the results and having all these nations be dependent on us and the turnaround time needing to be so quick.
It was not easy, but we got it done. We were really pleased that we were able to have the capacity in place before that happened.
Somehow, a lab for doing research that slowly expands the pool of human knowledge had been transformed into a public health lab, doing applied research that answered life or death questions.
It was really quite gratifying to know that all the effort we had put in over the years had come to a point where we were useful, something you can have an immediate application. I still love basic research, right? It's not like I'm going to switch over to applied research. But I think it was gratifying to know that the work you've done over the years is proving, useful.
So when we report results and then you hear about policy changes that would have most likely would have been impacted by work that you would have contributed to, information you would have contributed to, that's always it's very exciting.
Christine's work during the pandemic is just a small part of her contribution to the field of science and technology here in Trinidad and Tobago. She has been conducting and supervising research on viruses from dengue to chikungunya for 28 years. But her curiosity and interest in science date back to her childhood.
We'd sit at the dining table, you ask a question, anything. My father would leap up, rush into the study, get an encyclopedia. If he didn't have the answer, get the encyclopedia, and then they'd be reading the encyclopedia at the dining table. And if he couldn't find the answer in the dining table, or mommy didn't have it, he would call somebody.
Her father taught at the University of the West Indies, and her mother was a biology teacher at St. Augustine Girls High School. Christine, her parents, and her two sisters lived in university housing on a short street jam-packed with children.
The children just came and went. People would come, drop off children. The families were close. So it was a sort of communal raising of children. And so that's why it got the name the kibbutz, because it was, children being raised by any parent, any adult who happened to be there. So
she was fascinated by biology and read the textbooks for fun. When her mother taught her about viruses, she formed an interesting connection to science fiction.
In the books and in the movies, these UFOs would come and land and, take over, take control, take me to your leader and all the rest of it. And first, there's a virus. It's actually a virus that infects bacteria. It's called a phage bacteria, bacteriophage. It looks like a little spaceship. And it lands on a cell, and it injects its genetic material into that cell, and that genetic material has information that allows the virus to basically take over the cell as a factory to make more viruses. So this to me it just reminded me of those all the sci-fi. It was like a little alien invasion of your body. And so that's what initially fascinated me.
She won a national scholarship. Then it was off to King's College at University of London to study biotechnology. She found that virology was her favorite course, and her notes were coveted by fellow students. She studied hard, but never had to cram.
I was just organized. I always had a timetable. So in my study timetable for exams was, a couple of months in advance. Theirs was the week before exams. So I you know, went out to parties, went to museums, picnicked. I lined with friends, but I did my work. I would, you know, I think most days I would come home, I would go through my notes and stuff, but I never was up all hours in the night. You know, so I never pulled an all-nighter. Maybe something wrong with me, I don't know, all my friends were doing it, but I didn't.
Christine's PhD in molecular virology was conducted under the supervision of world-renowned retrovirologist Professor Robin Weiss. Weiss was famous for figuring out how HIV got into cells. But when Christine interviewed with him, she was blissfully unaware of his reputation.
At the end of the interview, he said to me, well, you know, I think that you'd be a good fit in the lab and I'm interested, and so we'd like you to join the team, right, as this PhD student. And I told Robin, well, I'll have to get back to you. I do have other people to see, and, you know, because I didn't know who he was.
Fortunately, she soon found out who Robin Weiss was and grabbed the opportunity to work with him.
And it was fabulous. It was a nice team. Robin was a great supervisor. I loved the way the team was very productive. But there was no, sometimes you get these research teams, there's a lot of internal competition and backbiting and politics and things. I didn't feel any of that. We had a great time.
Soon, she had notched over 8 years of study in the United Kingdom. With a PhD and over a year of postdoctoral research under her belt, she could have chosen to live and work in one of the world's big cities, but didn't.
I left Trinidad when I was 19, and, you know, I did not want to become one of those people who lives outside of Trinidad for 30, 40 years and then comes back and complains about how everything has changed. I wanted to experience being an adult in Trinidad. I wanted to experience working in Trinidad. And so I was happy to come back.
Besides, she acknowledged that she owed the government a period of public service in exchange for her national scholarship.
I once told my mother, when I got the scholarship, I said to Mommy and Daddy, you know, y'all are very lucky, you know, y'all don't have to pay for me to go to university. And my mother very quickly corrected me and said, I pay taxes, right? So she did pay for me. And I realized that the rest of Trinidad and Tobago had paid for me to go and do the studies. A lot of people used to run away from the service, but I wanted to come back and do it. I wouldn't have felt good if I didn't.
Christine was back home and looking for a job to fulfill her public service when she found a job posting for a lecturer in biochemistry at the University of the West Indies. She was just 26 years old.
I had not taught before. Obviously, I'd given lectures as a researcher, you know, I'd given lectures and stuff, but I had not taught before. But I come from a long line of teachers, right? So I don't know, teacher must be in my blood. So I wasn't totally nervous when I came back to do the lecture. I was pretty confident. That confidence was shattered at my first lecture when I totally mistimed how long it would take to deliver this lecture.
The students wrote her up on the bulletin board, asking if a 50-minute lecture could actually be completed in 35.
That same first class, when I walked in to teach, there was a mature student there. I don't know how old she was, maybe in her 40s. And she let out one long stoops when she saw me come in. She was like, please, I've paid my money, and this is what is coming to teach me, some little girl. And so, but we got along well afterwards.
She soon adapted to the demands of lecturing, but it was research that she loved, and supervising PhD students who were doing research. She had one student who was studying mosquito-borne viruses.
I had a student who used to go hiking, you know, used to be in the forest like 4 times a week, hiking an hour and a half with 50 pounds on his back, going setting up mosquito traps, coming back the next day, collecting the mosquitoes, and then we'd screen them to look for viruses in them. This is where dengue came from, Zika, chikungunya. They all emerged from animal, and SARS-CoV-2 emerged from animal populations, cross over into human populations.
And after mosquitoes, Christine supervised research on viruses in bats and monkeys, both potential vectors of disease in humans. After accepting her Anthony N. Sabga Award, Christine went home to Axel and Mia, 2 of the people who cheer her on, but also keep her solidly grounded.
You're under scrutiny now, so you have to do, you can't just get the award and rest on your laurels. You have to, you know, it's a mid-career award. So you want to, I mean, as an individual, I want to live up, continue to live up to it. So it does spur me on.
I can't think of anybody who's more deserving than Prof Carrington. Truly, and I'm not just saying that because she's my supervisor and because, I have such great loyalty and affections for her and towards her. But we were doing the math just recently, and she's been working in Trinidad in research, building her research career for going on 28 years now. And it's been 28 years, and I've been a part of that for quite a bit of it, and I've seen it. It's 28 years of just good, honest work. And she truly believes in the work.
It was an immense honor. I think it has given me more visibility and attracted more interest in the work that we do and also the work that the university does. I think that the awards in themselves is sort of identifying Caribbean excellence and rewarding it and sort of showcasing it is very important because it basically shows us that what we are capable of, shows not just as individuals but as a region, what we are capable of. What variant we seeing still?
All Omicron, and the majority of them are B5.2.1.
Okay. I was happy, not just for me, but for my team, because the work that we've done, not just on SARS-CoV-2, but over the years, it's truly a team effort. It's a community of people who worked hard together, and basically I felt as if I was accepting the award on their behalf. I could not have done it on my own, and it's not just the people in the lab. It's my parents, my other family, friends. I have a whole bunch of cheerleaders and people who encourage me and people who keep me very, very solidly grounded.
During the pandemic, Christine reached out to scientific collaborators in her global network to share findings and ask questions. She finds the global community essential to researchers, especially those working outside of the established centers of research.
When you're in a small setting such as ours, it's very easy to lose confidence. You sort of, you don't have, it's not like a huge research team and other research teams nearby that you can easily access, certainly before the internet. I mean, now the internet is here, it's easier. But, you can get isolated. And when you get isolated, scientifically isolated, you begin to question yourself. Is what I'm doing good enough? How does it compare to other people? And then you go to these conferences and you have your little work there, you put up your poster or you're giving a talk, and you realize that you ain't doing too badly. And people are coming and they're interested in what you're doing, asking questions. Then you realize that you're contributing and you're part of a bigger network. And I think that is it's so important to remain connected with the rest of the scientific world.
Now that the lessons of the pandemic have hopefully been learnt, Christine says her modest lab needs an upgrade, so we're more ready for disease outbreaks of the future.
It needs to be more efficient, we need to be more agile in the way that we do things. And I would really love to have some sort of research center, a research lab that can focus on sort of pathogen genomics and infectious disease research. And not just on the research itself, but also on training people in, people engaged in the public health bodies, so that the next time this happens, we are much better prepared. The lab we're in now, it's cramped, it's overcrowded, it's not fit, truly fit for purpose. And we need a home for that capacity and to build on that and to really move things forward, not just for Trinidad, but for the entire Caribbean, for the region, I think it's important.
The Anthony N. Sabga Awards, Caribbean Excellence, are presented annually. Nominations are received from all across the Caribbean, candidates are screened, and winners are presented. These are mid-career awards, and the cash prize is meant to acknowledge a body of work, as well as spur more of it. We congratulate our 5 2022 laureates and look forward to receiving many more nominations from around the region in 2023.
This is what Professor Holmes, Edward Holmes, an Australian research fellow and a leading evolutionary biologist and virologist, and who is also noted for having co-authored the publication of the first genome of SARS-CoV-2, had to say about Professor Carrington. Every region needs a local virologist hero to steer them through the COVID-19 project and pandemic. And Christine has played this role brilliantly in the Caribbean, and this has literally saved thousands of lives.
The Office of President also represents and reflects the national pride and joy that we Trinbagonians feel when one of our own is placed in an appropriate and well-deserved spotlight. Professor Carrington is one such individual. She has been quietly and without ceremony working diligently in the field of molecular genetics and virology, a national authority on the subject, respected far and wide. She first came to national attention as a member of the UWI's COVID-19 task force. Her role in understanding the epidemiology of COVID-19 and related viruses in the Caribbean cannot be overstated. She has also become a familiar and confidence-imparting face on our television screens over the last 2 years, explaining to us laymen the significance and implications of COVID-19 and other viruses. It is therefore with great pleasure that I this morning present Professor Carrington with her well-deserved award.
There is an old saying, if you want to go fast, go alone. But if you want to go far, go together. I believe in together. I have always traveled with a village, a posse, a group. And I have been happiest as part of a team, a couple, a community. And it is to that village, that community, that country of engaged citizens that I belong. I recognize and thank the University of the West Indies, which has been part of my life since I was a child. My father was a staff member, and UWI was my playground. The institution and so many individuals there supported and facilitated the development of the infrastructure and expertise that allowed us to respond when our region was in need. It is never easy in a setting such as ours. Sometimes it's two steps forward and one step back. For me, those 2 steps forward were possible with the advice, support, collaboration, and friendship of colleagues like Professors Dan Ramdath, Michel Montai, Abiodun Adesion, Jonas Adai, and Chris Ora. I have found fulfillment with my little team of ever-resourceful and hard-working graduate students, postdocs, and technicians, scientists, researchers, seekers of truth who have pushed me to continue learning and who keep me young. I can't name them all. It's been 26 years at UWI. But today, I want to recognize and thank Dr. Nikita Sahadeo, Mr. Vernie Ramkisoon, Ms. Anushka Ramjag, Dr. Ariane Brown-Jordan, Mr. Soren Nichols, and Mr. Nicholas Mohammed, who supported the fight, the ministry, the country, and the Caribbean in the battle against COVID-19.